Hi guys. For this week, we're going to start looking at all the classes of eukaryotes that we haven't looked at yet. There's only 5 weeks left, so we're not going to look at all the classes of eukaryotes. We're going to follow the ones that are in your textbook. So that includes euglenoids, cryptophytes, cryptomonads, hepatophytes. Those are the ones for this week. And hopefully we'll stay on schedule for the rest of the semester. Okay, so what do you need to know for this week? What do you need to know for your quiz and just in general? Well, I want you to understand how we think secondary endosymbiosis occurred. We think there's at least 2 lineages. And I want you to become familiar with the groups in the green lineage. Now, these in your textbook are considered to be in the green lineage. New data shows that they're actually not all in the green lineage, but like I said, we're following your textbook. So for the purpose of this quiz and all other quizzes, I will always defer to the information in your textbook, even though if it might not be completely up to date. Okay, so you've seen this before a million times. We started with the Archaeoplastida. Primary endosymbiosis, right? There was only really one event. There was that other event with the Polynella, which nobody really talks about all the time. Really, there was one major event and one very minor event. But for secondary and tertiary endosymbiosis, it's not so clear. So I'm going to walk you through what we think happened. Okay, so just for a little review, secondary and tertiary endosymbiosis, you know this already, right? Here is primary endosymbiosis, and here is secondary endosymbiosis, right? Eukaryote eats primary endosymbiosis event, and tertiary is it eats the secondary. I mean, it's pretty obvious, but I just want you to all know what it's about before we go in forward. So we're going to be talking about these organisms for the rest of the class, the ones that are the result of secondary or tertiary or quaternary, who knows? Okay, so now, as you guys know from those week's readings, it's widely accepted that secondary endosymbiosis has happened. What's not known is how or when exactly. We don't really know the beginnings of it, but as you can see from this figure, there's the green lineage. So everything in this, everything in this gray circular area, these are thought to have been the result of secondary endosymbiosis. And you can see that there's a green lineage that happened by organisms eating an Archaeoplastida, or there's a red lineage from organisms eating a rhodophyte. Now remember, rhodophytes are the ones we talked about a long, long time ago, right? The red algae that are also in the Archaeoplastida. So let's start. Now, the names that are in this figure aren't exactly the names that are in your text. Don't worry too much about it. I will tell you which ones you need to know about, okay? Like I said in the beginning of this lecture, we're not going to go through all the groups. We're only going through the ones that are in your text. So we're not going to talk about all the ones in this figure. Okay, so let's just talk about the green lineage. 2 events. So secondary green plastids erode independently in the euglenids and the chlorioacniophytes. Now if we go back here, there's chlorioacniophytes and the euglenophytes. Your textbook only talks about the euglenophytes, so we're only going to talk about them today. But these are both the product of an organism from the chlorophyte being engulfed by a heterotrophic protist. I already showed you that. Okay, so that's the green lineage. So really 2 events, but in the red lineage, it's not so clear. So sorry, I'll move my face. The red lineage, we've got the cryptophytes, the heteroconophytes, the hepatophytes, and then a whole bunch of other stuff, which we're not going to talk about today. But here's all I want you to know is that we think, in one theory, it's a free-living red alga, a rhodophyte, an archaeoplastid, was captured and later diversified into multiple lineages, right? So all of these groups came from one common ancestor. That's one theory. Another theory is that, and this is that theory, right? There was this one secondary endosymbiotic event with a rhodophyte, and then from that organism we get all these different existing lineages. Okay, so in this theory, all of these guys are monophyletic. But in the other theory is that they're not monophyletic, that all of these groups, the cryptomonads, the padophytes, the straminopods, the dinoflagellates, and the apicomplexans, are not monophyletic, they're polyphyletic, and they resulted from multiple secondary endosymbiotic events. This isn't resolved, or even it could even be horizontal gene transfer. This isn't resolved, we don't know yet, but I want you to be aware that this is unresolved. We really don't understand how many times in secondary endosymbiosis occurred. We don't yet know. And in fact, there are some examples that it's still in progress. So this organism called Tatina arenicola that was discovered very recently. I think it was 2009 on a beach in Japan. Is it a plant? Is it an animal? It's such a bizarre creature. So this organism, heterotroph, captures an alga, and you can see in this picture, right? There's a clear organism. It captures the alga. The heterotroph divides, and only one daughter cell keeps the alga cell. The other daughter cell is free living and can survive and hunt until it captures an alga cell, and at which point it becomes photosynthetic. So it seems like this is an example where it's secondary symbiosis in progress. Now the difference here, of course, is this algal cell is a fully functional cell. It hasn't lost most of its organelles yet. Come back in, a million years, who knows? Maybe less than a million years. Who knows how long this takes? Okay, what do we know for sure about secondary endosymbiosis? Well, we know that all primary plastids, the archaeoplastids, probably arose from one endosymbiotic event, but there's two, because don't forget about Polynella. Everyone always forgets about Polynella, but almost all the archaeoplastids came from one event. But the secondary plastid organisms, we don't know. We don't know. It could be one. It's at least two. We know here it says one or several, but it's two or more subsequent events, because we know for sure there had to have been at least two. Okay, so by the end of the class, I'll want you to be familiar with all these 7 supergroups. But like I said, in this class, in your text, we're only going to talk about these groups, right? And they're representatives from each of the, we're going to talk about representatives from each of these groups. We're not going to talk about all of them. We just don't have the time or the space. So we've already talked about Archaeoplastida. Today we're going to talk about some secondary endosymbionts in Archaeoplastida, although this is a misclassification, I just want to remind you. And we're going to talk about Excavata, which includes euglenoids. Okay. But before we get into too many details, I just want to talk about these supergroups and how they relate to eukaryotic evolution and protists in general. Protists are found in all of these groups. Some of the groups are only protists. So when you think of what's a eukaryote, we always think of animals or plants or fungi, but eukaryotes are almost entirely protistan. Some of the groups of multicellular life, but all of the groups have protists, and some of them are only protists. So it's a really important distinction. Some of them that are only protists are alveolata. That's only protists. Straminopla, only protists. Rhizaria, only protists. Excavata, only protists. Amoebozoa, only protists. So the only ones that aren't exclusively protists are Opisthocanta and plants. So definitely the minority. Okay, so here's a slightly different version of that same phylogeny. It's the occurrence of chloroplasts among eukaryotes. Okay, so algae with primary plastids here, and also where are the euglenids? It's not exactly the same, so it's a little bit confusing. I don't know where it is, but you could include euglenids somewhere around here. But here's where all the organisms are that have secondary plastids. So they're kind of all over the place. It's not as neat and tidy as the primary plastids. So like everything in evolution, it's kind of messy. But hold on, it gets even messier. There's some non-photosynthetic groups. Completely, they've lost the ability to photosynthesize. But then there are some groups that have secondary plastids that are mixed in with the archaeoplastids, right? And they're not actually mixed in with the archaeoplastids, they're just misclassified. And then there are some organisms over here that are actually primary endosymbionts, or that are mixed in with the secondary plastids. So long story short, there has been some horizontal evolution of secondary endosymbiosis. It's not clean. Okay, that was a really messy way to say that. Okay, so the paper this week is talking about the early evolution of the early eukaryotes, and we haven't really talked about that in this group, and there's a reason for that, is because we don't know very much about them. We know there's bacteria, and there was eukarya, or archaea, and then suddenly there was eukaryotes, and there's all the supergroups that we know about today, but what was before the supergroups? What were the earliest, very, very earliest archaea like? Were there any pre-mitochondrial eukaryotes? We talked a little bit about this, but not in any detail. Now, there is a group that's been proposed called Archaeozoa. It includes Giardia. You've probably heard of Giardia, Trichomonas, and Hexamida. Now, is this a real group? Is this a fake group? These organisms are all parasites, which kind of makes sense. If you don't have a mitochondrion or any plastids, you're gonna have to borrow them from your hosts. But here's the thing. They probably lost their mitochondria secondarily, and they're probably not even closely related, because if you guys have taken any evolutionary ecology, there's this phenomenon called long branch attraction, where groups, if you do a phylogeny, groups that evolved a very long time ago appear to be closely related, even though they're not. And it just has to do with how the trees are constructed. So they appear to be closely related, but they're really not. So we know that Archaeozoa are not a monophyletic group, but they're not, some of them are ancient, but they're not all closely related. So we still don't know that much about the very earliest eukaryotes, this little group in here. Okay, so let's talk more about what we do now. Euglenoids, right? We've talked a little bit about these because we talked about the discovery of all the membranes, the 3 and 4 membranes in the chloroplasts. But euglenids are they're not just euglenids in this group. There's trypanosomes and leishmanias, and these are both parasites. So they look like euglenids. So trypanosomes causes a disease in humans called trypanosomiasis, and it's an African sleeping sickness. And there's 2 kinds of it. It's carried by flies. But there's also leishmanosis, which is a skin disease. And it's also caused by an organism that looks a lot like Euglena. They all look very, very similar. So it's kind of creepy to think that you've got these organisms living in your body. These are all parasites except for the euglenoids. You know a little bit about euglenoids. You might not know there's over 1,000 species. They're freshwater. A lot of them, 2/3 of them are non-photosynthetic. So they've lost their plastids, and they're all unicellular with one exception, but they've got a huge range of different kind of morphologies and body sizes. They're really, really kind of charismatic little organisms. I don't know if they're charismatic. I think they are. But what's important about these organisms, I think, is they've really blurred the line and made people think very differently about eukaryotes. There's not just animal, plant, and other. This is a very artificial distinction, and really these distinctions describe habits, not states of being. They're just different ecological habits based on conditions, right? And so Euglena can hunt or can photosynthesize depending on conditions and whether or not it has plastids, of course. So eukaryotes exhibit a continuum of lifestyles, and it depends on the pressure of the environment. What's also very interesting about euglenoids is they were discovered a long time ago by Van Leeuwenhoek. This was a Dutch microbiologist. He invented the first microscope, and he was the first person to describe microbes, and he described Euglena. So like 400 years ago, we knew about Euglena, and he said, I mean, he made these fantastic drawings of all these crazy little creatures he saw with his microscope in the 1600s. I saw a great multitude of living creatures in one drop of water, amounting to no less than 8,000 or 10,000, and they appear to my eye through the microscope as common as sand does to the naked eye. I mean, look at these pictures. They're pretty fantastic. And you can imagine how exciting it was for him to look at a drop of water and see these amazing creatures. It would have been like magic. I mean, it seems so second nature to us now to think, oh yeah, there's microbes, but they didn't know that. They didn't even know these creatures exist. They looked like little beasts. Anyhow, I just think that's really cool. I love his pictures. Okay, another thing that's important about the euglenoids is they don't have a cell wall. They have a pellicle. And this is, the plasma membrane is just supported by protein strips in the cytosol, and it gives them a huge amount of flexibility to change shape, as is obvious in this video, which is not working. I don't know why it's not working. Anyhow, they contract and expand really wildly to move around. I'm sorry that it's not working. You can always look it up. There's a lot of videos on YouTube about Euglena swimming around. Okay, and these guys, there's never any end to how cool they are. They never have sex, not ever. Not ever. Or maybe not. Maybe they're just really discreet and we haven't discovered it yet. But so far, no one's ever documented sex in Euglena. And you'd think they would. We've known about them for 400 years. Maybe they evolved before sex. That's very possible. But they have, or maybe they just don't need sex. They could have lost sex. I mean, I love to talk about how these traits are lost if they just don't need them. Isn't that something to think about? What kind of environment would you have to live in that you didn't need sex anymore? Something to think about. Anyway, so what happens is the nucleus duplicates, and then the flagellar processes, the gullet and the stigmata, they start separating. And then longitudinally, it just splits, the whole cell splits in half, and you have two identical daughter cells. I mean, seems pretty efficient. I don't know, maybe it's painful, but it seems to work pretty well for these guys. And some people drink or eat Euglena as a nutrient, as a vitamin, really. I don't know. I don't know much about that, if it's good, if it's not good. Never had it, but it's out there. Actually, I think I should try it. Why not, right? Okay, that's all I want to talk about euglenids. Let's talk about cryptomonids. Crypto is, the prefix crypto means hidden. Now, I just want to re-emphasize here again, your textbook puts them in the Archaeoplastida, but molecular evidence doesn't put them in here. We know they are somewhere else. I'm not exactly sure. I don't want you to worry about it. But what's important is they didn't come from the green lineage. They came from the red lineage, right? We talked about two different lineages of secondary endosymbionts. These guys came from the red lineage, even though they're green. Remember, you don't have to be red to belong to the red lineage. You just need to have chloroplasts that were inherited by a rhodophyte. There's about 200 species. They're aquatic and marine. They're very tiny. They have photo and phagotrophy, like a lot of organisms. But these guys do have sex, and they're pretty darn cute. I mean, look at them, the little plated bodies zipping around. Like I said, the phylogeny is unresolved, even though there's very clear evidence that they have secondary endosymbiosis. They used to be classified as a Stromanopula. They have been classified as an Archaeoplastid, but we're not sure. We know now that they are in the red lineage, but not in, oh yeah, it is in this figure. It wasn't in the last figure, but it isn't, it's not in this figure. It was in the last figure. Okay, and there's a little picture so you just get an idea of what they look like. That's only, that whole organism's only 10 microns. So they're very, very tiny. And here's, this is from your textbook, but you can see the nuclear morph, right? That is residue from the second organism that was engulfed by the heterotroph, whatever that was. And the endoplasmic reticulum and the chloroplast. I don't know why. And one more thing about these guys is these guys are a source of a new industry. The phycobiliproteins, and we've talked about those are the accessory pigments in chloroplasts. They're pretty stable when they're extracted, and they are, look at those colors, they're beautiful. So these are being cultivated, farmed for their phycobiliproteins, and they're used in cosmetics, as dyes, and all sorts of things. So it's, they're so pretty. Okay. Haptophytes. Hapto means binding. And again, these guys are put in with the plants in the Archaeoplastids, but they are also from the red algal lineage. They're not separate from that. These guys are really cool. They're these little tiny plated, kind of like tank-like organisms. Here's only 1 micron, right? That scale bar is only 1 micron, so they're extremely tiny. They're unicellular. They're not flagellated, and they have these crazy wild plates all over their bodies, and that's what makes them so cool. They also have a haptonema, which is not a flagella, right? You can see the 2 long flagella and a haptonema in the middle, and that's like a little, like a little probe, and helps them maybe with hunting, with navigating. We're not exactly sure what it does, but they all have it, and it's like a little appendage. But like I said, the most important thing about them is their coccoliths, and these are, their outer membrane is covered with plates, calcium carbonate plates, and they are highly decorated and highly beautiful. Not 100% sure what they're used for, but likely to improve buoyancy. Some people think that they help concentrate light. But what they do is they leave an excellent fossil record. So there's a lot of evidence of these organisms in the fossil record, and you can tell a lot about the climate and the geology and just what was going on Earth because of these guys. And there's some more examples. One weird thing about these guys is they are responsible in part for cloud nucleation. So during algal blooms of these guys, haptophytes become aerosolized. So they are either ejected through wave action or they evaporate into the atmosphere and serve as a nucleus for cloud formation. And one of the reasons they're so good at this is because of their shape and because of their buoyant coccoliths. They stay in the air longer than other organisms. And yeah, I mean, the connection between these microscopic marine creatures and weather patterns, like who would have thought? I mean, I say these things as like they're so normal, but they're just wild. This one type of organism is responsible for cloud nucleation. But they're also responsible for these big foamy beach events. I don't know if you guys have ever been to the ocean and the beach looks like there's just been a big shampoo event, big shampoo tanker that's crashed. This is some a glycoprotein from coccolith colonies. Phaeocystis. It's polysaccharide embedded in mucilage and floating colonies, and it's just an algal bloom. That's it. It's not like it's not anything terribly toxic, but it's kind of interesting. Okay, that's it for this week. I don't want to overload you with details about organisms. This is just an attempt to get you familiar with interesting, cool little organisms that live all around us, and some of them live in us too. All right, I will talk to you later.